Hi, I'm Ismaël Koubiyr, and I'm a postdoctoral fellow at the Neurocentre Magendie in the University of Bordeaux. And today, I will talk about my PhD thesis work regarding the dynamic of modular-level alterations of structural functional coupling in clinically isolated syndrome. We know that the brain can be modeled as a complex network of structurally and functionally interconnected regions, where each region is represented as a node and each connection as an edge. And using diffusion tensor imaging, or DTI, we can map the structural connectivity between different gray matter regions using white matter tractography. On the other hand, functional connectivity will represent the synchronization in activity between pairs of gray matter regions. In MS, abnormalities in both modalities have been previously reported. It has been shown that functional connectivity can be shaped and constrained by the underlying anatomy. Also, the structural functional coupling, which represents the association between functional and structural connectivities, was demonstrated to be an important parameter allowing to detect more subtle brain alterations in other neurological diseases. However, whether this structural functional coupling could be altered in the early stages of MS is currently unknown. So, in line with the literature and due to the nature of MS, we hypothesized that the disruption of connections may lead to a structural functional decoupling. And in order to test our hypothesis from the early course of MS, we combined functional connectivity and structural connectivity to investigate the longitudinal evolution of both global and modular specific topology changes in the brain in the first year following a clinically isolated syndrome. And for that, we used a cohort of 41 CIS patients and 19 matched health controls. Each participant was scanned using a 3T MRI, including anatomical sequences, resting state fMRI, and DTI. Additionally, they were also evaluated using an extensive neuropsychological battery. After processing our resting state fMRI and diffusion data, we registered them to our parcellated anatomical sequence to obtain functional and structural connectomes. Also, we parcellated our brain to different modules to look at modular alterations. Then, using graph theoretical metrics, we looked at the global level reorganization of our brain. And finally, we assessed the structural functional coupling as the Pearson's correlation coefficient between strands of structural and functional networks. It is important to notice here that this correlation concerned only direct structural connections, meaning that we looked at the functional connectivity between A and B only if there was a direct anatomical connection between them. If the connectivity between A and B was going through a third region C, this connection was not considered. When looking at our population characteristics, we can see that our CIS patients have a very low T2 lesion volume at both time points, with the median EDSS score of 1 as well, indicating a low disability at this stage. Now, when looking at the cognitive performances of our CIS at baseline, they showed some significantly lower Z-scores compared to the healthy controls in the CSCT test, which is an information processing speed test similar to the SDMT, and also in the brief visual-spatial memory test. However, after 1 year of evolution, these differences were not present anymore, and our CIS showed similar performances compared to healthy controls. Regarding functional connectivity, we found no difference between our CIS patients and our healthy controls in the overall network metrics or the modular level metrics. However, after 1 year of evolution, we noticed an overall brain functional network reorganization as betweenness centrality was significantly higher in CIS compared to healthy controls, as well as some modular level alterations in different parts of the brain. When looking at the structural connectivity network at baseline, global brain damage was already present as the clustering coefficient was significantly higher in CIS compared to controls in different regions of the parietal, occipital, and temporal lobes. Modular level alterations were also present with significant modifications in both intermodular and intramodular connections. However, after 1 year, there was no more difference between CIS and healthy controls in the overall structural network metrics, but modular level alterations were still present at this stage. Regarding our structural functional coupling, it was preserved in CIS compared to healthy controls at both the whole brain and modular levels at baseline. After 1 year of the disease onset, the whole brain coupling was still preserved. However, when looking at the modular level, we noticed a preservation of the structural functional coupling in the default mode network, the frontal parietal network, but the decoupling appeared at this stage in the salience, visual, and somatomotor networks. Regarding the clinical correlates, we found that both the MSFC score and the CSCT performances were strongly associated to the structural clustering coefficient at baseline. So, to conclude, this longitudinal study showed that structural damage may precede functional reorganization in the first year following a CIS, and importantly, we showed that structural functional decoupling is only present after 1 year of the disease onset, and specifically in the salience, visual, and somatomotor networks, along with preserved cognitive performances at this stage. So finally, all these results suggest that functional reorganization that may be responsible for the compensation mechanism at this stage is probably occurring along indirect anatomical pathways. And our future goal is to further follow up these patients after 5 years of their disease onset and try to better understand the neural correlates of cognitive functioning in the early stages of multiple sclerosis.